Hi everyone. In this lecture, we'll talk about a novel virus, SARS coronavirus 2. As we know, the COVID-19 pandemic that began at the end of 2019 has changed people's life. Up to now, novel coronavirus infections have resulted in over 78 million confirmed cases and over 1.72 million deaths globally. Before we start novel coronavirus, let's look at coronavirus first. A human coronavirus was isolated for the first time from the nasal secretions of a male child with a common cold in 1965 by Tyrrell and Bino. Because of their morphological similarity with a solar corona and an electron microscope, crown-like, the virus was termed coronavirus. Coronaviruses are enveloped 120 to 160 nanometer particles that contain an unsegmented genome of single-stranded positive sense RNA, the largest genome among RNA viruses. The genomes are polyadenylated at the 3' end. Isolated genomic RNA is infectious. The helical nuclear capsid is 9 to 11 nanometer in diameter. There are 20 nanometer long club or petal-shaped projections that are widely spaced on the outer surface of the envelope. The viral structure proteins include a phosphorylated nuclear capsid M protein, a membrane M glycoprotein that serves as a matrix protein embedded in the envelope lipid bilayer and interacting with the nuclear capsid, and the spike S glycoprotein that makes up the petal-shaped peplomers. The genome organizations of a representative coronavirus is shown in figure. The SARS coronavirus genome is about 29.7 kb. Boxes shaded in yellow represent open reading frames, ORF, encoding structural proteins. Boxes shaded in lavender encode non-structural proteins. The separate ORF within each gene are translated from a single mRNA species. S is for spike, E is for envelope, M is for transmembrane, N is for nuclear capsid. Characteristics used to clarify coronaviridae include particle morphology, unique RNA replication strategy, genome organization, and nucleotide sequence homology. Coronaviruses form the subfamily, also Coronavirinae, which is one of two subfamilies in the family Coronaviridae, order Nidovirios, and realm Riboviria. They are divided into the four genera: alpha coronavirus, beta coronavirus, gamma coronavirus, and delta coronavirus. Alpha coronaviruses and beta coronaviruses infect mammals, while gamma coronaviruses and delta coronaviruses primarily infect birds. There are 6 coronaviruses that can infect humans. The alpha coronaviruses 229E and NL63, and the beta coronaviruses OC43, HKU1, SARS coronavirus, and MERS coronavirus. This picture shows the transmission of coronaviruses. Coronaviruses can cross species barriers and cause severe diseases in humans, such as MERS and SARS. Coronavirus is just an example of a string of pathogens that have come from wildlife trafficking, including SARS, Ebola, bird flu, and many more. The wild animals can harbor diseases that can make other animals, including humans, sick. Wildlife trade markets facilitate viral transmission. They allow multiple species to be in proximity that otherwise would not come into prolonged contact with one another, which could be a factor for violent strains of pathogens spreading to humans. Additionally, human capture of wildlife and incursion into natural wildlife habitats allow viruses to jump the species barrier. Let's look at the coronavirus replication cycle. Virions bind to specific receptor glycoproteins or glycans via the spike protein. Penetration and uncoating occur by S-protein-mediated fusion of the viral envelope with the plasma membrane or endosomal membranes. Gene 1 of viral genomic RNA is translated into polyprotein, which is processed to yield the transcriptase replicase complex. Genomic RNA is used as a template to synthesize negative-stranded RNA, which are used to synthesize full-length genomic RNA and subgenomic mRNAs. Each mRNA is translated to yield only the protein encoded by the 5' end of the mRNA, including non-structural proteins. The N protein and the newly synthesized genomic RNA assemble to form heliconucleocapsids. Membrane glycoprotein M is inserted in the endoplasmic reticulum (ER) and anchored in the Golgi apparatus. Nucleocapsid N plus genomic RNA binds to M protein at the binding compartment (ERGIC). E and M proteins interact to trigger the binding of virions, enclosing the nucleocapsid. S and HE glycoproteins are glycosylated and trimerized, associate with M protein, and are incorporated into the maturing virus particles. Virions are released by exocytosis-like fusion of vesicles with the plasma membrane. Virions may remain absorbed to the plasma membranes of infected cells. The entire cycle of coronavirus replication occurs in the cytoplasm. In December 2019, a pneumonia outbreak was reported in Wuhan, China. On 31 December 2019, the outbreak was traced to a novel strain of coronavirus, which was given the interim name 2019 nCoV by the World Health Organization (WHO), later renamed SARS coronavirus 2 by the International Committee on Taxonomy of Viruses, and the WHO named the disease caused by the virus COVID-19. SARS coronavirus 2 has been identified as a new strain of beta coronavirus with approximately 70% genetic similarity to the SARS coronavirus. The virus has a 96% similarity to a beta coronavirus, so it's widely suspected to originate from bats as well. SARS coronavirus 2 is a positive single-stranded RNA virus that is contagious in humans. The single-stranded RNA genome of SARS coronavirus 2 was 29,891 nucleotides in size, encoding 9,860 amino acids. Similar to other beta coronaviruses, the SARS coronavirus 2 genome contains two flanking untranslated regions, UTR, and a single long open reading frame encoding a polyprotein. The SARS coronavirus 2 genome is arranged in the order of 5' replicase (ORF1A or B), structural proteins, spike S, envelope E, membrane M, nucleocapsid N, and 3'. The S glycoprotein and the transmembrane glycoprotein M are two major envelope proteins. The S glycoprotein is an antigen that binds to the receptor and is responsible for cellular fusion. M glycoprotein has a role in envelope formation and virion assembly. Phylogenetic relationship among SARS coronavirus 2 and other beta coronaviruses. The genome of SARS coronavirus 2 has overall 89% nucleotide identity with beta SARS-related coronavirus, SL, CoV, VXC21, and 82% with human SARS coronavirus, BG01 2003, and human SARS coronavirus TOR2. Although the degree of diversification of SARS coronavirus 2 is lower than that of, for example, influenza viruses, the divergence of two prevalent evolvement types of SARS coronavirus 2, L type and S type, was reported. According to this study, strains in L type derived from S type are evolutionarily more aggressive and contagious. It's clear now that SARS coronavirus 2 also uses ACE2 as a cellular receptor to infect humans. SARS coronavirus 2 is sensitive to ultraviolet radiation and heat, and can be inactivated by lipid solvents, such as diethyl 75% ethanol, chlorine-containing disinfectant, peracetic acid, and chloroform. SARS coronavirus 2 can be inactivated for 30 minutes at 56 centigrade. The transmission of respiratory droplets and mucous membranes infections are the main route of SARS coronavirus 2. The SARS coronavirus 2 mainly spread from person to another through small respiratory droplets from the nose or mouth, where a person confirmed COVID-19 coughs or exhales. These droplets land on objects and surfaces around the person. Other people may catch the virus by breathing in droplets or touching these objects or surfaces, then touching their eyes, nose, or mouth. SARS coronavirus 2 can be transmitted through the nose-eye, possibly through the way of increased oral pressure caused by coughing or sneezing, and reverse transmission of the virus through the nasal lacrimal duct to the diaphragm and then infect the conjunctival cornea. The human angiotensin-converting enzyme 2, ACE2, known as the major receptor for the virus S protein, provides the entry point for SARS coronavirus 2 to capture and infect a wide range of human cells. ACE2 is found in many types of cells and tissues, including the lungs, blood vessels, heart, liver, kidneys, and gastrointestinal tract. It's also present in the epithelium, lining the lung, the nose, and mouth. Regulating blood pressure and inflammation are the main functions of local ACE2 via the conversion of angiotensin II to other molecules that neutralize the effect of angiotensin II. Occupying the ACE2 receptor by SARS coronavirus 2 prevents it from performing its normal function and breaking the angiotensin I and angiotensin II peptides. Naturally, there is a high concentration of ACE in the lung tissue. Thus, in ACE2 deficiency, ACE will be more active due to more available angiotensin I, which is changed into angiotensin II. Increase the local angiotensin II levels, damage blood vessel linings, and cause inflammation and tissue injury. This picture shows the replication cycle of SARS coronavirus 2 in virus-susceptible host cells. ACE2 binds to the receptor binding domain of the spike proteins, allowing for fusion with the host cell membrane. Positive single-stranded RNA is released, partially translated into SARS coronavirus 2 polymerase protein and transcribed. The resultant subgenomic RNA translational S, M, and E proteins are taken to the host cell ER membrane and later combined with nucleocapsid protein N. Post-Golgi processing or elements are incorporated into a mature virion and trafficked to the cell membrane for exocytosis of newly budded SARS coronavirus 2 particles. After SARS coronavirus 2 enters into the lung from mouth and throat and infects the cells, the virus replicates in the cell and releases more new viruses. Massive accumulation of SARS coronavirus 2 leads to a surge of immune cells and pro-inflammatory cytokines, which resulting in a rapid increase in cytokine levels in the blood or releasing more virus particles into the blood circulation. The virus and cytokines positively induce high expression of ACE2 in the intestinal epithelium and other organs, which accelerates the overexpression of ACE2 and viral binding, causing the systemic infections with the virus. Okay, we have talked about the structure and pathogenesis of SARS coronavirus too. We'll continue with the virus next time. See you soon.